Good morning, students. Hope you all are fine and had gone through the last lecture. Today we will start with translation. Under translation, we will go through steps in translation, differences in prokaryotic and eukaryotic translation, regulation of translation, post-translational modification, and inhibitors of translation. We will cover this topic in two sessions. In first session, we will see the requirements and steps of translation in eukaryotes. Now, learning objectives. After this lecture, you should know what is translation, what are the basic requirements for it, and stages of translation. So, first definition. As the word translation means to translate in a language which can be recognized or understood. So it is the pathway of protein synthesis and the language of the nucleotide sequence on the mRNA is translated into the language of the amino acid sequence. In eukaryotes and prokaryotes, translation stages are almost same with few differences. So here we will see the translation for eukaryotes and compare it with the prokaryotes. Now the requirements of translation. First, mRNA to be translated. Second, tRNAs. Third, ribosomes. Fourth, energy in the form of ATP and GTP. Fifth, enzymes and specific factors. So, mRNA, it carries genetic information. In eukaryotic mRNAs are monocistronic and prokaryotic mRNAs are polycistronic. Now the tRNAs. tRNAs, these are bilingual or they have dual role. That is, it reads the codon on mRNA and then supplies the corresponding amino acid to form polypeptide. Third, ribosomes. We can say these are the work benches, means where the translation occurs. So, ribosomes in prokaryotes it is 70S and in eukaryotes it is 80S and they have two subunits, one the larger and smaller. In prokaryotes, the larger one is 50S and smaller one is 30S, and in eukaryotes, larger subunit is 60S and smaller subunit is 40S. So, large subunit has separate site for elongation factor and peptide bond formation, and smaller subunits have G-coding site where the tRNA anticodon and mRNA codon interaction occurs. And ribosomes also have E site, P site and A site. E is the exit site, P is the peptidyl site and A is the amino acyl site. Now the steps of protein synthesis. Activation of amino acids is the first step, then initiation, elongation, termination and the last one is folding and processing, that is post-translational modifications. So first, activation of amino acids occur or the charging. First, the amino acid is combined with AMP and it forms amino acyl adenylate and which is attached to the corresponding tRNA and it forms the amino acyl tRNA. So activation occurs. Now, the second step is initiation. Initiation has four sub-steps. That is first, ribosomal dissociation. Second, 43S pre-initiation complex formation. 48S initiation complex formation and 80S initiation complex formation. So, what happens in initiation? Before the protein synthesis, starts in initiation, one of the steps. It involves the assembly of the components of the translation system before the peptide bond formation occurs. And these components include two ribosomal subunits, mRNA, amino-acyl tRNA, and GTP and initiation factors that facilitate the assembly of this initiation complex. But there is difference in initiation factors in prokaryotes and in eukaryotes. In prokaryotes, there are three initiation factors which are known as IF1, IF2 and IF3. While in eukaryotes, there are at least 9 initiation factors and they are designated as EIF to indicate the eukaryotic origin. So, in initiation, first step is ribosomal dissociation. Here we can see the inactive 80S ribosome of the eukaryote. It has 60S and 40S subunit. So, ribosome splits into the smaller 40S subunit and larger 60S subunit. EIF3 and EIF1A binds to 40S subunit and stabilizes the 40S subunit, and to the 60S subunit EIF6 binds and stabilizes the 60S subunit. Now second step is formation of 43S pre-initiation complex. So in 43S pre-initiation complex, what happens? As methionine is the starting amino acid in eukaryotes and formylmethionine is in prokaryotes. So activated methionine, that is with initiation factor 2 and GTP, it forms the ternary complex and it binds with the stabilized 40S subunit. So in this picture you can see the ternary complex is with the mTRNA plus eukaryotic initiation factor 2 and GTP. This comprises ternary complex which attaches to the stabilized 40S ribosomal subunit, and 43S pre-initiation complex is formed. Now, the third step in initiation is formation of 48S initiation complex. So, for it, there is one step, that is the mRNA activation, which occurs in eukaryotes only and not in prokaryotes. It is with the help of the 4F complex. 4F complex is composed of 4G, 4E and 4A. So the whole 4F complex is a cap binding factor. 4E recognizes cap of mRNA, 4A removes secondary structure of mRNA, and activated mRNA binds to 43S pre-initiation complex, which is mediated by EIF3 and EIF4. So 48 as pre-initiation 48 as initiation complex then scans the mRNA in 5-3' direction for initiation of codon, and 5-AUG codon is surrounded by Kozak sequence. So we can see the initiating tRNA recognizes AUG codon along with Kozak sequence in eukaryotes. In prokaryotes, the starting amino acid is formylmethionine and starting tRNA recognizes the AUG codon along with Shine-Dalgarno sequence. So, the difference in sequences in eukaryotes and prokaryotes, that is Kozak sequence in eukaryotes and Shine-Dalgarno sequence in prokaryotes. Now, the formation of ATS initiation complex occurs. The eukaryotic initiation 5' factor EIF5V. Here you can see it facilitates the binding of 60S ribosomal subunit to 48S initiation complex and 80S initiation complex is formed. At this point, methionine tRNA is on P side of ribosome to start the elongation. Here you can see this is E side, this is P side and this is A side. So P side is the peptidyl side where tRNA with methionine is located and A site is amino acyl site where the incoming tRNA charged with next amino acid is placed and E site where the uncharged tRNA after donating its amino acid exits the ribosome. So, the initiation completed with the formation of 80S initiation complex. Now, the next step is elongation. It is also a three-step process. It involves several proteins called as elongation factors. First step involved in elongation is binding of amino acyl tRNA specified by the next coding triplet in the mRNA to the A site. Second is formation of peptide bond and third is translocation. So, when the binding of amino acyl tRNA to P and A site after the formation of fully active initiation complex ATS, the amino acyl tRNA methionine is already positioned at P site as we already see. And EF1A, that is elongation factor 1A, places the coming amino acyl tRNA to the A side with the use of GTP. Now the second step is the peptide bond formation. Peptide bond is formed when NH2 group of amino acid in A site carries out a nucleophilic attack on the carboxylic group of the tRNA in P site. So peptidyl transferase catalyzes the peptide bond formation and the peptide bond is formed here and shift of methionine to amino acyl tRNA on in A site. Third is the translocation of ribosomes. After peptide bond formation, the tRNA in the P site is empty. Its amino acid is bonded to amino acid of tRNA in A site. Here we have seen that this amino acid is bonded with this one. So, elongation factor 2 after complexing with GTP binds to tRNA on A site and displaces it to P site, pushes tRNA from P site to E site from where it exits the ribosome and repeated cycles takes place. So, peptide bond formation and elongation continues for multiple cycles in a polypeptide synthesis. Now, after elongation, the third stage is termination. And this elongation continues until the stop codon is encountered. There are three stop codons, UAG, UAA and UGA. So, in this figure, we can see as the stop codon is encountered and releasing factor recognizes the stop codon, it hydrolyzes the peptide bonds with the peptidyl transferase. The peptide the bond between the polypeptide and tRNA in P site. It is hydrolyzed and this releases protein and after that dissociation of ribosomes occur. So this releases protein and tRNA through the exit site. Upon release of protein, 80S ribosome dissociates into 40S and 60S subunits by recycling. And 4 ATP of energy is used in formation of each peptide bond. So that's all about translation steps and stages in eukaryotes. We will continue in the next session. Hope you can understand. Thank you.